Welcome to Have You Read It, the channel where we bring books to life, one summary at a time. Today, we're diving into David A. Sinclair's fascinating book, Lifespan: Why We Age and Why We Don't Have to. Sinclair, a leading geneticist and professor at Harvard Medical School, doesn't just explore the science of aging. He challenges our very understanding of it, arguing that aging is a disease, and a treatable one at that. Sinclair's journey begins with a fundamental question: why do we age? He challenges the traditional evolutionary theories that suggest aging is simply a matter of natural selection, waning after our reproductive years. While acknowledging the contributions of scientists like Medawar, Williams, and Kirkwood, Sinclair posits a bolder, more unifying theory: the information theory of aging. Drawing on his decades of research, particularly his groundbreaking work with yeast, Sinclair illustrates how aging is not primarily about the accumulation of genetic mutations, as previously thought. He explains how cloning disproves this theory since healthy animals can be cloned from old cells, showing aging is not necessarily because of irreversible changes to our genes themselves. Instead, he focuses on the epigenome, the control system that dictates which genes are turned on or off. This system, he argues, is where the crucial information loss occurs. This is where his research with yeast becomes so illuminating. Yeast cells, surprisingly similar to human cells, also age. Sinclair's lab discovered that as yeast cells age, they accumulate extrachromosomal ribosomal DNA circles, or ERCs. These ERCs, he explains, cause genomic instability, distracting essential proteins called sirtuins from their regular duties of maintaining the epigenome. This distraction, he proposes, is the root of aging, a process he describes as epigenetic noise. To test this theory, Sinclair's team created genetically modified mice, dubbed ice mice, whose DNA could be intentionally damaged without causing mutations. The results were striking. The IC mice aged prematurely, developing various age-related conditions, despite no alteration to their underlying genetic code. This experiment, he explains, shows how epigenetic noise, not genetic mutation, drives aging. But if aging is a loss of information, can it be reversed? Sinclair emphatically believes so. He points to examples in nature, such as the bristlecone pine and the bowhead whale, whose lifespans dwarf our own, as proof that aging is not an immutable law. He also details his own research, showcasing how activating sirtuins with molecules like resveratrol and NAD boosters can not only slow aging, but even reverse some of its effects in mice, such as improving vascular health and even restoring some aspects of fertility. Sinclair doesn't just confine himself to the theoretical. He outlines practical steps anyone can take right now to engage their longevity genes. Eat less. Calorie restriction, or intermittent fasting, has consistently shown to increase lifespan and health span in various species. Sinclair recommends periodic fasting for maximum benefits. Lifespan also warns about high consumption of meat and dairy and the role of excess amino acids, particularly methionine, on the activation of the mTOR enzyme, which is linked to accelerated aging. Move more. Exercise, especially high-intensity interval training, activates longevity genes, improves cardiovascular health, and lengthens telomeres. Sinclair highlights research showing that even short bursts of intense exercise can significantly reduce all-cause mortality. Embrace temperature extremes. Exposing yourself to hot and cold temperatures can activate longevity genes. While more research is needed on cryotherapy and sauna use, Sinclair emphasizes that our bodies benefit from being outside the thermoneutral zone. Beyond lifestyle changes, Sinclair discusses promising pharmaceutical interventions like metformin, rapamycin, and NAD boosters, all of which have shown positive results in animal studies. He also discusses emerging technologies like senolytics, which target and eliminate senescent zombie cells, and cellular reprogramming, which has the potential to reset the epigenetic clock and reverse aging at a cellular level. Sinclair's own lab has achieved remarkable results with reprogramming in mice, restoring vision and nerve function in aged animals. The future, as Sinclair paints it, is full of both immense potential and significant challenges. While optimistic about the prospect of dramatically increased lifespans, he addresses the potential social, economic, and environmental consequences of a world where people live much longer. He advocates for increased public funding for aging research, rethinking our social safety nets like Social Security and pension systems, addressing the ethical concerns around gene editing and longevity interventions, finding innovative solutions to consumption and waste, ensuring equitable access to life-extending technologies, openly discussing death, and giving individuals the right to choose when and how they die. Lifespan concludes not with a prediction but a call to action. It's an invitation to participate in a conversation, not just about how long we live, but how well and how we, as individuals and as a society, can shape a future worth living in. It's about engaging with our own longevity, not just for ourselves, but for future generations. The main takeaway is that aging is a disease, and we can treat it. We are not bound by the limitations of our evolutionary past. By understanding the underlying mechanisms of aging, we can make choices today that impact not just our own lifespan, but the future of our species. Thanks for watching. Make sure to give this video a thumbs up and hit that subscribe button so you never miss our next book dive.